give people a chance to nominate.
You all stand up and do jumping jacks and we'll warm up.
You start.
Actually, this is a great opportunity. Smile. I'm trying to get both. Thank you.
So let's go ahead and get started. So thank you all for joining us. We got a good crowd here. So now we're gonna have Gabrielle Exelic join us. She's doing her research during the summer at University of Chicago, and we're excited to hear about your summer project. So take it away.
Awesome. Hi, everybody. I'm Gabby, and I've been working in the Overlab at the University of Chicago this summer. And my presentation is the qPCR validation of asthma-associated genes differentially expressed in airway smooth muscle cells in response to interleukin 13 and interleukin 17. But before I start off, has anyone ever used like an inhaler before? Like, raise your hand. Don't be shy. Well, my fellow asthmatics, you are not alone, because asthma affects nearly 25 million people in the U.S. Asthma is characterized as inflammation of the airways, as well as airway hyperresponsiveness, and it is genetically heterogeneous. Now, airway hyperresponsiveness is a hallmark feature of asthma. Airway hyperresponsiveness is the contraction of the airways, predominantly airway smooth muscle, in response to environmental stimuli like pollen or air pollutants. So over here, you can see that the muscle is relaxed, and that's before the exposure to the environmental stimuli. And over here, you can see that the muscle is very contracted, and that's after the exposure to the environmental stimuli like pollen or air pollutants. So airway smooth muscle circumferentially surrounds the bronchi. The bronchi of the lumen. So over here we have a cross section, so you see that the airway smooth muscle is surrounding the lumen. And let's blow up airway smooth muscle cells. So you're probably wondering, hey, what are those little blue dots like around the cell membrane? Well, that is a cytokine. And a cytokine is a pro-inflammatory mediator that's associated with asthma pathogenesis. So the way that this works is cytokines bind to the airway smooth muscle cell receptors, and then the membrane receptors signal to the cell nucleus, ultimately affecting the gene expression of the airway smooth muscle cell. Two cytokines that we were looking at this summer and are very important involved in asthma are interleukin 13 and interleukin 17. And interleukin 13 is a mediator in airway hyperresponsiveness, and it induces hypercontractility. Interleukin 17, not only does it induce hypercontractility, but it also, the severity of airway hyperresponsiveness is positively correlated with IL-17 expression levels. So my project this summer is based on a larger study done by the Overlab. So what they've done is they've taken airway smooth muscle cells from 75 donors obtained through the Gift of Hope organ and tissue donor network. They were cultured in the presence or absence of IL-17 and IL-13, and after 24 hours, RNA was collected and for genome-wide expression studies. And from those studies, we have obtained, they have obtained lists of response genes that have been enriched for asthma genes. So you see right here, that's for IL-13, IL-17, and they're both in combination, and And my project this summer is to validate their gene expression, validate that SMAD3 is downregulated and ORMDL3 and RORA are upregulated. And I will do that using qPCR. So what is qPCR? Well, qPCR stands for quantitative polymerase chain reaction. And the greatest part is we can we can find the concentrations of the amplified DNA in real time, which is really awesome. The qPCR gives us cycle threshold values, or CT values, which are the number of cycles to detect a fluorescent signal. And using the average CT values, we can find the efficiency, which is the ability of the primers to detect the gene product. Now, why are we using qPCR? Well, it's quick, it's efficient, and the qPCR results can validate that RORA and ORMDL3 are upregulated while SMAD3 is downregulated for the gene expression. So to get the ball rolling, we need to make some cDNA. So we use the superscript 3 first-strand synthesis system for RT-PCR. Then we need to find a control gene, because an efficient control gene is essential for qPCR. Control gene is used as a standard to compare to that the test genes are working well, and we were like, yeah, this is great. Then we have our control gene, we have our test genes, so we design primers and then we optimize them. And by optimization, we mean running them through a temperature gradient, as well as running through the products through a gel to see the optimal temperature they work at. And then we find the efficiency. Again, the efficiency is the ability to detect the gene product, like the primers detecting the gene product. And we're using dilutions of cDNA. And we run the reaction at the optimal temperature we found through the optimization stage. So currently, we have all of our test genes. We have all of them successfully optimized. We found a control gene after long trial and error. We have really good efficiencies, which will lead us into the next step of our project, which I will discuss in the next slide. So over here, I have two gel images. One is of ORMDL3's gradient, and these two are RORA and SMAD3's gradient. We compared to a 100 base pair DNA ladder. So over here, right here, you can see that these genes are around 100 base pairs. And the optimal temperature that worked for all three test genes was 61.8 degrees Celsius. And over here, you see we can see a primer efficiency graph of SMAD3 in our control gene. The average CT value is on the y-axis, while the log dilution factor of our cDNA is on the x, and that's how we find our efficiencies. So the next step is we need to perform a qPCR. When I get back to Chicago from here, we're performing a qPCR with our test genes in addition to our control gene, and we're going to use DNA synthesized from 30 different asthmatics. And we're going to do this, and if we get the products that we're looking for, we validate these genes, and we can say that, oh yeah, SMAD3 is downregulated, ORMDL3 and RORA are upregulated. And the next step is to test other genes on these lists, because it's important to validate these, because airway smooth muscles relation to asthma is largely unexplored. And it's not only will we, gain a better understanding on airway smooth muscles relation with the cytokines, but asthma as a whole. So I would like to thank Dr. Carol Over, my PI, Dr. Emma Thompson, my mentor, the Over Lab, the Fire Program through the Leadership Alliance at UChicago, the Office of the Provost, and the RISE program at Beaupre College. Thank you very much. Me and my mentor just looked at the list. We were like, all right, let's just start with these. We didn't really have a specific process in choosing which ones, because eventually we want to just keep validating all of them exactly. Yes?
I was wondering what kind of asthmatics you got your samples from?
Awesome. Okay, well, I have an additional slide for that. We got the range of the samples we got. The median age was around 49 years old. We have a ratio of men to women, and we have European Americans and
and what do you think is the next step? The next step to follow that?
Well, once all of, you know.
Once you have
our-
an expression profile, you know, this goes up, this goes down, what's next?
Well, we kind of have like, you know, a better understanding of how cytokines are affecting like the gene expression in neuroendocrine muscle. So I would think that we would just keep focusing on more of the like effects of the cytokines rather than, you know, just enriching for asthma genes, just more of the relationship between the areas of the cytokines. Maybe test different cytokines as well, in addition to IL-13 and 17.
Does that have treatment implications? Is that what you're saying?
Treatment?
Yeah, I mean, in terms of dealing with asthmatic conditions.
Yes.
How would it matter?
I think I wrote this. Well, you know, like, I'm not entirely sure, but go back. Okay. Go back to your list. Okay, here we go.
So we all
Oh my
know,
goodness.
so all of these may have some role. Is there anything that can be done if you know that a particular gene is upregulated? Is there any kind of medication you can create? Is that something that the overlap may want to collaborate with someone on?
It's definitely like a goal, but I'm personally not sure what they would want to do with that. But I know that it is one of their aims to like eventually get to some sort of treatment.